We live in a crowded world, 2 million kinds of living things occupying every imaginable place. A seashore, home to strange, exotically beautiful life forms. A high mountain snowfield. Here live vast numbers of tiny organisms. Red pigments shield their delicate cell machinery from intense ultraviolet radiation found at high altitude. They are well adapted for this Mars-like environment and live here without competition from other life forms. At the mountain's base is another kind of extreme environment, a hot spring. Near the boiling point, living things abound, but they are very primitive, microscopic forms of life. The field of biology that investigates why organisms live where they do and how their populations interact is ecology. Ecologists may study the distribution of organisms in space and time. Or they investigate broader questions. How does energy flow through a living community? How are materials shuffled around within an ecological system? Others focus on single populations. What affects them? What do they affect? At any of these levels, ecology becomes a complex and fascinating inquiry into the interconnected threads that bind all living things. Feeding, competing, capturing, or being captured. Along an isolated coastline, the shifting tide washes over the rocks in its daily cycle. Here is a fantastic natural laboratory for investigating how populations interact with each other and with their environment. Mussels, for example. Why do these common shellfish form a dark band along the wave-washed coast? Looking at the sharply defined mussel beds, an ecologist starts asking questions. Could food limit the mussels? More information is needed. What do they eat? Microorganisms filtered from the sea water. Wherever waves wash over the intertidal rocks, food is available. So why are the mussels limited? Food must not be the answer, at least not the whole answer. How important are predators? Birds can get at the upper mussels and dine at leisure while the tide is out. How about competing species? Barnacles cover the rocks just above the mussel beds. Could barnacles limit the mussels by occupying their growing space? Perhaps the delicate mussel larvae cannot survive on the hot, dry rocks above protection of adults. Experiments have shown that all of these factors can play a role in governing the mussel population's upper border. But the lower border is another problem. Almost everyone who visits the sea has seen starfish dining on mussels. Could predation by starfish limit the lower border of the mussel bed? Studies have shown that starfish cannot tolerate as much exposure as mussels. These slow-moving predators must live well below their prey. As the tide comes in, they creep at imperceptible speed to the lowest mussels, feed, and return before receiving too much exposure to sun and drying air. Evidence that starfish limit mussel distribution came from a bold experiment in which all starfish were removed from a stretch of coastline. Result? The lower border gradually extended, in 2 years doubling the size of the mussel bed. The nice thing about studying mussels and starfish is that they hold still. You can count them, remove one species or the other, and check the effects. However, predator-prey interactions are not always this easy. How many mountain lions live in this canyon? How many deer? You can search for days and not see one of these secretive animals. Avoiding such difficulties, some ecologists have taken the problem of interacting populations into the lab and put it under the microscope. Meet Didinium. Its name means terrible nose, with good reason, for this is a predator as ferocious as any which spring from the imagination of science fiction. Didinium is a microscopic wolf with an appetite for only one kind of prey, the smoothly swimming, slipper-shaped paramecium. These scenes, photographed in natural time, show an awesome example of predation. A predator swallows a prey larger than itself. In 30 minutes, it will take another, and then another. After engorging, Didinium divides, producing new individuals that will repeat the ravenous process again and again. With such an efficient predator at large, its prey should be brought to extinction. Yet paramecium continues to be one of the most abundant organisms in lakes and ponds. How is this possible? Such questions can be answered through experiments that allow populations to interact in a controlled way. To begin, a few Didinia are added to a culture of paramecia, and daily counts are made of both populations. By day 6, Didinia are becoming abundant in the culture. On day 7, a few paramecia are left, hiding in the bushes, and there are a great many Didinia. Dinner is over. On day 8, an interesting change. Every Didinium in the culture has developed a shell-like covering that shows up brightly under polarized light. Within its protective shell, a Didinium enters a state of dormancy, suspended in time. Its predator temporarily out of the way, paramecium's population builds up until, responding to chemical signals, a Didinium comes out of dormancy and the feasting begins all over again. This relationship has been going on cycle after cycle for untold millions of years. Like starfish and mussel, the association continues because in its natural environment, predator cannot eliminate its prey. However, if paramecia are cultured in a clean dish, a simplified environment with no microscopic bushes or fish skeletons to hide in, the Didinia will totally consume their prey. They compete for the very last paramecium, ending a cycle and bringing about their own extinction. Only in a complex environment, one with bushes and organisms to get in the way, can paramecium and Didinium coexist. Laboratory studies of this kind bring up questions about our own ecology. For example, humans depend upon a highly simplified environment for food. Single crops grown on huge factory-like farms. In order to understand our own system, ecologists would like to know more about natural systems. The Earth itself is a giant ecological system. Sunlight streams in, energy for life. Carbon dioxide, water, nitrogen, oxygen, raw materials flowing in great cycles between atmosphere and living world. Ecologists call this global life support system the biosphere. Within the biosphere are more or less distinct ecological units called ecosystems. A seashore is one kind of ecosystem, a pond another, forest, desert, grassland. Each system has its own character, yet in basic ways, these major ecosystems are alike. For example, all run on solar energy, as plants, the food producers, convert light into the chemical energy of food. This process, photosynthesis, occurs in tiny green bodies within plant cells. Using light energy, these chloroplasts manufacture essential nutrient molecules. In go raw materials: carbon dioxide, water, nitrates, phosphates, and sulfur compounds. Out come carbohydrates, fats, proteins, nucleic acids, the molecules of life. When an animal eats a plant, it picks up a supply of biological molecules. Molecules that can be used in two ways: for energy to drive its metabolism, and as a source of materials for building its own products. What effect can these molecular level events have on ecosystems? Studies show that only a small fraction of energy contained in one organism is transferred to the next consumer. Most of the energy picked up in food is used in activity and lost as heat. Because of this low transfer of energy, an ecosystem can be expected to have a greater mass of producers than consumers, and more plant eaters than predators. Amount of growing area required to support a human population will depend upon what they choose to eat. If this plot supports X number of people eating plant foods, a much greater area would be required if these crops were fed to animals and animal products were eaten instead. As long as the sun shines, energy for life is renewable. But what about raw materials used by plants to synthesize food? Nitrogen, for example. Imagine a nitrogen atom contained in a protein, part of a plant cell. In winter, when the plant dies back, the protein is digested by a bacterium, and our atom becomes incorporated into a bacterial protein. The bacterium is eaten by an earthworm, and the nitrogen atom is returned to the soil in the worm's waste products. Picked up by a root hair, our nitrogen is again incorporated into plant protein. Animals eat plants and in turn are eaten or die. Nothing is wasted as borrowed molecules return to the system through the activities of scavengers and decomposers. Shuffling molecules is a vitally important ecological process. If raw materials are not kept in circulation, an ecosystem runs down and supports less and less life. This is exactly what happens when range animals go to market. They carry away borrowed molecules and the range becomes less productive. Recycling is what keeps natural ecosystems running. However, modern agriculture is an ecological system that for the most part does not recycle materials. The crop is removed. Its molecules pass through our bodies and are then deposited in a sewage treatment plant. From here, they go into a river or the sea, but in few cases are they returned to the field to nourish new plants. Instead, fertilizer made with a high energy cost is brought in to ensure next year's crop. How long can this forced system of food production continue to work? Another important question the new field of human ecology must try to answer. Ecology is a relatively new science, but its findings already carry an important message. Humans are part of Earth's life support system. How we fit our activities into the great cycles of the biosphere, and how we choose to interact with other living things will determine the quality of life from here on.